let's talk Ediacaran biomolecules. Rare, hard to find, hard to pin down, but there's one case, pretty famous now, very recent, where they seem to have done that. Hmm. So, shown here are things that are like Dickinsonia, we think. They all have what they call glide symmetry, which is not bilateral symmetry, but symmetry that's offset, where you make the same thing, but you do it up and to the left, and then up and to the right, and then up and to the left, like that. So, yeah, not quite bilateral symmetry, although it's drawn here as if it is. But you can see here on the Andiva, it definitely isn't. Here's a Yorgia at the bottom. It's got some kind of big digestive gland at the top by the look of it, along with a glide symmetric body. Andiva, glide symmetric, but it looks like it's being melted like Salvador Dali clocks. And here's a Dickinsonia, just a little round disc. These are all Ediacaran. You find them, Dickinsonia is pretty common in the Ediacara hills. Andiva and Yorgia and some Dickinsonias are actually found in the White Sea region in Russia, around Arkhangelsk. Saying that badly. But there are, around that region in Russia, there is occasionally, it is possible that you get a different preservation. So all of the other ones, including all of these, it's the death mask preservation. Sand turns to sandstone, creature is gone, and you have a sandstone mold, like that. But in one case, you get something like this. There is a fossil, Dickinsonia, as I live and breathe. It's kind of squished, so it's there was a three-dimensional Dickinsonia, then the sand hit, and it got flattened a little bit, and then it dried onto the roof, and so it's mostly hollow underneath. There's a thin crust of organic matter that is horribly cross-linked and altered and all kinds of stuff, but actually preserves some of the original biology of Dickinsonia. And darn it, you can analyze that. These are very new things, but yeah, potentially possible. Here is organically preserved Dickinsonia, and C is an organically preserved Andiva, we think, that they were also going to look at. Here is the cleaned out version of the Dickinsonia fossil. You can see it. It's gorgeous. Oh my gosh, it's perfect, but it was better with the organic material in there. And what they're looking for here is evidence of certain types of steroid molecules. So there's cholesterol. That's the steroid that we find in, let's say, animals, metazoan animals, things like us. Then there is ergosterol, and that is a different steroid, different version of the steroid, has a different number of carbons, crucially, because we're going to do this by mass spec, where we can measure the mass. It's like the weight, but it's the mass of a molecule. And it's distinct from the stigma steroids, which are plant versions. They all are cell membrane molecules, and they're all very important to life in those. And they all start off from the same starting products. It's just that they are finished in different ways that make them distinctive. So you can tell material that has come from an animal from material that came from a fungus or a plant, for example. And so here we go. These are the amount of 27 carbon sterols and STEROL, like steroids, but the alcohol version. And you can see in the actual Dickinsonia, there is a bunch of that. It's actually really high. C27s are over 90% of the sterols you get there, whereas on the rock around them, you do get remnants, traces of organic matter, but it's less than 10% is these cholesterol type steroids, C27s. So that right there, that figure is the best evidence that Dickinsonia was an organism with animal-like metabolism. It may not be an actual animal, but on the way toward that. Here's looking in more details at some of these. So here's Dickinsonia. The Andiva actually did not show up very well in this, although it was organically preserved. the steroids come out a little different, or there's not as much. So it's perhaps also along the path to becoming an animal, but it doesn't finish its steroid biogenesis in quite the same way. So even though Andiva and Dickinsonia kind of look similar, they may not actually be that close to each other if we're thinking of steroid biochemistry as kind of a defining characteristic of what is and isn't animal-like. Yeah. So there we go. These are these are some of the data. Stigmasterine is a 29 carbon version of a steroid, and that one is from various algae mostly, but plants as well. But although this is way before we have the first plant fossils, so definitely be thinking algae. There's your cholestains, which are your C27s, and those are what you get in animals like cholesterol type molecules. And they're differentiating the type, the ratios of those two things. And within each of those categories, they can differentiate the particular branch points on the molecule. Molecules are similar, but there are differences, and they can be detected. So here we go. Typical preservation for Ediacaran biota macrofossils is totally dissolved, death mask, fossil is gone. But in this one place, these couple of fossils, there is actually what appears to be organic carbonized matter in there in the fossil. Within that, they can find evidence of cholesteroids specifically in the Dickinsonia. It's unclear why the Andiva did not show up with exactly the same profile, which you would have figured. It's from a little bit different place. Did the things not fossilize right? Did it finish its steroids differently? Anybody's guess. How can we tell them apart? Molecular fossils, count the carbons. C29 plant, C28 fungus, C27. There you go, cholesterol. How does mass spec work? That's Google it. Google it. Yeah. Based on steroid composition, Dickinsonia is animal-like in its biochemistry, in its steroid biochemistry. And I think that's all you can really safely say. But it makes it closer to animals than to anything else, and that blows a lot of wild fan theories out of the water as to what this could have been. So there you go.